Dr. Jim Pearle has been practicing here in Fullerton, in Northern Orange County for 38 years. And he's a graduate of the University of Chicago, where he did his undergraduate. He did his medical school at New York Medical College. He did his residency at George Washington University in Washington, D.C., and his pulmonary fellowship at the University of Illinois Medical Center in Chicago. He has been the assistant professor of medicine in the Division of Pulmonary Disease at UCLA School of Medicine. He was the director of bronchoscopy, which is a procedure in which pulmonologists look down into your lung for diagnosis and treatment. He was assistant professor of medicine in the Division of Pulmonary Diseases at UCI School of Medicine. And he has been the director of respiratory therapy in multiple hospitals in Northern Orange County, including St. Jude, Martin Luther, Whittier, several hospitals in Northern Orange County, and he's done that. He's also American College of Chest Physician member, American Thoracic Society, and very important, he's a St. Jude Medical Center Institutional Review Board, which means that any study that's done at St. Jude Medical Center has to be reviewed and okayed and approved by this board, which is made up of people like Dr. Pearle. He's been a participant in more than 150 drug trials over the last 20 years. He's been published in multiple research and medical journals, and he's been in active practice here for, as I said before, 38 years. So please welcome Dr. Jim Pearle.
Well, for me, there are a couple of firsts for giving a talk. You know, I've been giving talks on COPD and asthma and respiratory problems for 20 or 30 years, and I can kind of wing one of those at the drop of a hat. But this one's a little different, talking about clinical trials, even though I've spent about half of my professional life over the last 20 years doing clinical trials. It's really the first time I've had a chance to give a talk. So a couple of remarkable things. You're going to see me with a few note cards because some of this is a little new to me. And number two, you'll see me with glasses so I can see the note cards. So we all got a bunch of firsts. Okay. When we are reduced to mere observation, science crawls. Scientists said that in the 1800s. It's kind of food for thought. What do you think he means by that? If all we do is observe and don't control the environment, how do we draw conclusions? How do we learn things? We might learn things, but we learn them slowly, and we learn them randomly. So once we know that, it understands why science has moved in a direction of trying to do research and do trials. And what do we really mean by a trial? Anybody got a definition of a clinical trial?
I know, you try medicine on some people and placebos on another.
Well,
what happens?
That's a pretty fair description. A good kind of precise one would be a prospective, organized, systematic exposure of patients to an intervention of some kind, whether it's drug, whether it's surgery, whether it's dietary, and observe the results. So you want it to be systematic and you want it to be organized. That's how you come up with data. Now, and as somebody else said, this should be under the investigator's control, subject not to data availability or chance, but to his ability to ask questions and design means of answering them. So that's sort of where the thought of doing clinical trials. Now, in ancient times, the Hebrews, the Babylonians, the Egyptians took pretty good care of themselves. They kept the environment clean. They had some things that were medicines, the remedies to treat different problems. But the information from them was pretty much gained by chance, by observation, and not by controlled information. And in ancient times, even the advanced civilizations said no real science is how they collected this information. But when do you think the first clinical trial was? Any ideas?
World War I?
A little earlier than that. Let's go earlier than that.
Civil War?
Okay, we're not going to get far enough back. In the book of Daniel, Nebuchadnezzar, King Nebuchadnezzar, had young men of royal lineage, and he thought that they would be strongest and best if they were on a diet of meat and wine. Now, Daniel wanted the sons, and some of the others didn't want to eat meat and wine. They wanted to eat bread and vegetables. So Nebuchadnezzar allowed one group to eat, to stay on the meat and wine diet, and the other group to be on the bread and vegetable diet. And surprisingly, after a couple years, the ones that were on the bread and vegetable diet were actually healthier and stronger. And Nebuchadnezzar concluded that he wasn't going to be dogmatic about keeping the royal men on the meat and wine diet. So there's our first clinical trial, 500, 600 BC. But there weren't many more. Now, in around 1025, a medical writer named Avicenna suggested just what you described, that you do a couple of different groups and you try some different remedies on them and you see how they turn out. But there's no record that that trial was ever actually done. Most of the time, what people were doing was what the Babylonians did. What they did is they put their sick people in public places so that onlookers could look at these people and they could give their own ideas about how you might treat them. Not very scientific, but maybe they came up with occasionally some pretty good remedies. But that doesn't really fly very well. So many more years went on until finally in 1537, Ambrose Pearle, who was a surgeon assigned to combat, treated wounded soldiers and treated their wounds with boiling oil. And that was his standard remedy. I mean, it worked. It sterilized the wounds, but it was pretty miserable. So he ran out of boiling oil, and he tried a remedy of yolk of eggs, oil of roses, and turpentine. And he put that on some of the soldiers in their wounds, and he didn't sleep well that night or for several nights because he wondered if his remedy just might kill him. As it turned out, they all did well. And he said to himself, well, I'm never going to go out and subject these guys to boiling oil again. I'm going to use other remedies. So we're starting to get a little feeling of how science is slowly getting to be applied. But the next good clinical trial wasn't for about another 200 years. In the meantime, people were trying the usual haphazard remedies. There's a diarist named Samuel Pepys in England who reported the first paid clinical subject where a local college hired, quote, a poor and debauched man to have sheep blood let into his body. He did okay. So that's sort of a trial, kind of an observational trial, but at least it's a trial, but that's the state of kind of what they were doing. But the next most important trial came about 200 years later. James Lind was a British naval physician. And if you remember back in the 1700s, what disease was the curse of scurvy? Nobody at the time realized what caused scurvy, and the mortality rate of scurvy on these ships was terrible. And when Lind heard that at least one kind of semi-round-the-world ship trip, about three-quarters of the crew died, which is pretty staggering. So on his next trip, he planned a little trial. He took 12 men with scurvy, because they were all getting scurvy, and he gave 2 of them a quart of cider. He gave 2 of them some kind of elixir, meaning some kind of alcohol. He gave 2 of them seawater. He gave them 2 of them vinegar, and he gave 2 of them oranges and lemons. Pretty haphazard, pretty random. But the 2 who got oranges and lemons improved dramatically enough that one of them he actually assigned to be a nurse to the other men. This was a breakthrough, realizing that citrus and vitamin C was going to be the treatment for scurvy. Now, because providing citrus in one form or another to ships was expensive, there was a lot of resistance. So even though he published his data in around 1750 or so, it took about 40 more years before the British started making sure that there was citrus in one form or another on ships. But this was really a breakthrough trial. So trials kind of lurched along. In 1863, the middle of the 19th century, was the first time somebody used a placebo. Everybody knows what a placebo is, right? A placebo is some kind of inert substance that you use as a comparison to the drug or treatment that you're trying to study. So they first used placebos, and it's pretty important to use a placebo, otherwise you can't really demonstrate, you can't convincingly demonstrate that your product, that your drug really works properly. Now, we're making a little progress in this front, but at the same time, there was a lot of resistance to trials. The anti-vivisectionists didn't want trials or experiments on animals, and they kind of equated animals and humans together, so they didn't want trials on humans, they didn't want trials on animals. And in one famous quote, they really didn't want a physician, they didn't want a physician to be replaced by a scientist at the bedside. That was the climate at the time. The most notorious, and I guess really the famous of these anti-vivisectionists, shockingly, was the playwright George Bernard Shaw, who coined the phrase human guinea pig. New to everybody. So Bernard Shaw was a prominent anti-vivisectionist. But fortunately, things moved on, and despite this resistance, research continued. Now, but on the other hand, medicines were out there were lousy. There was no control. Nobody knew what they were being produced, but was being produced. In 1880, about 28% of all the drugs being made were so-called patent medicines. Now, patent medicines, by term, sounds really sounds like this is pretty serious, but all that was patented was the label. Nobody patented the products. And by 1900, 72% of drugs were patent medicines. So things were out of control. Only the label was patented. Nobody was looking at either efficacy or safety. So who jumped into the fray at about that time? Well, it was the AMA. So the AMA formed a council on pharmacy and chemistry. And they sort of took it upon themselves. It really was, the government wasn't really involved yet, and kind of took it upon themselves to give some kind of guidance. So they gave a seal of approval from JAMA, or the Journal of the American Medical Association, to drugs that that they knew something about the contents were reasonably safe, and that provided some kind of symptomatic relief. No proof of efficacy at all, but symptomatic relief. And that continued for about 50 years as the AMA, or the AMA, really JAMA, was providing this seal. And that was the best we knew for about drugs at the time. Then in 1906, the government passed the Pure Food and Drugs Act, which listed about 11 dangerous ingredients, among them being opium and alcohol. Now, once they started requiring and notifying, people became aware that the contents of a lot of these patent medicines had really damaging contents, the manufacturers started pulling back a little bit. So this started to at least improve the safety of the drugs, although it still didn't really tell us very much about efficacy. Even at that time, the Supreme Court and Congress failed to pass a law to prohibit manufacturers from making false claims about their drugs. Now, in 1911, in a famous court case, U.S. versus Johnson, we had Dr. Johnson with his product, Dr. Johnson's Cure for Cancer. And the Supreme Court finally began prohibiting false and misleading claims, kind of based on this Supreme Court case. Then, 1912, they added the Shirley Amendment, which started talking about fraud when manufacturers were fraudulently making claims about the contents and the benefits of their drugs. So as we kind of track along this time, clinical trials were becoming a little more sophisticated. They were becoming a little broad-based. And investigators were starting to do their trials cooperatively. So that the number of subjects that you had in a trial was larger, and statistically it was easier to be able to demonstrate significant results. In 1920 was the first time randomization really emerged. In other words, at least you made some effort to randomly assign patients to one group or another. Because otherwise, what you had investigators picking out their best subjects and their best patients to give their drug to, and then the second ones they give the placebo to, and sure, they'd show benefit. So it wasn't until the concept of randomization came along that you started to approach obtaining reasonable and valid data during these trials. Then, following randomization, the next step was blinding. And blinding meant that the subjects wouldn't know what if it was double blinding, the subjects wouldn't know what they were getting, and the investigators wouldn't know what they were giving. So there could be no bias from either the subjects who might behave differently knowing whether they were getting drug or placebo, or the investigators who might behave differently knowing whether they were giving drug or placebo. So we're starting to kind of remove all the bias from these trials.
What year was that?
The randomization was and blinding were 1920s, 1930s. But at the same time we got investigators working on that side, we still got some bizarre stuff going on because nobody is really enforcing the drugs. There is a product out called Banbar. I don't think anybody remembers Banbar, but it was meant to treat, this was in the 1930s, and it was meant to treat diabetes. What was in it? Milk, sugar, and horsetail. And it finally went to court because obviously this was really a lot of BS. So the Banbar people in this court brought out a bunch of testimonials about how effective this drug was doing. And what the government did is they pulled out a bunch of death certificates of pretty much the same people saying they died of diabetes. So finally, the Banbar was sort of exposed, but there still wasn't really a law to really take it off the market yet. Even as late as 1938 in this. It took a new law in 1938 to pass to finally finally be able to restrict these drugs. And this was also the first time that officials and people evaluating drugs could use data. They could use historical data. They could use uncontrolled trials. They could use other information rather than just the prospective trials in terms of assessing a drug. Still a lot of resistance to research. As of around 1934, the laws were generally that physicians had to practice in pretty much traditional manners. In other words, you couldn't do anything too out of the box. That's what you expected. But as of 1935, in a famous case, the Michigan Supreme Court said that if modern medicine is to progress, there must be a certain amount of experimentation. So finally, the government is starting to acknowledge that there's a science to this, and you have to take certain risks and certain steps to be able to develop new drugs. But it didn't. But not everybody kind of followed this. There was a drug called sulfanilamide in the 1930s. Sulfanilamide was a very good drug, and it was used to treat strep throat very effectively. So somebody decided to package sulfanilamide with diethylene glycol. Right, exactly. Pretty much antifreeze. And did no safety testing, no animal trials, no human trials, but they sold it and distributed it and killed about 100 people. Now the company, it was too much for the company, and by this time the FDA, the Food and Drug Administration, was getting a little stronger, so it took all the force of the Food and Drug Administration plus the company to retrieve this sulfanilamide. So as of 1938, the Food, Drug, and Cosmetic Act required safety data for development of new drugs. And now, as of 1938, that if a new drug was being developed, you had to send what's called an NDA to the FDA for the FDA's evaluation. And the FDA could sort of say yay or nay, but there was kind of but the default position at the time was really that the drug would be approved unless in 60 days they disapproved it. So the default position was still that those drugs could go on to market. And so we're seeing a little progress a bit at a time on this. But the FDA really didn't regulate and organize trials and methodology really till the 1960s. Now the first double-blind controlled trial was in 1943 in British office and factory workers with a drug called Pearle that was meant to be used for the common cold. It was a good trial. They did a double-blind controlled trial, but no benefit. Pearle failed. But at least they were starting to do those trials. Now, in 1947, after World War II, Scientists met in Nuremberg. You all kind of know about Nuremberg after World War II. They met in Nuremberg, and they came up with the Nuremberg Code, which helped us to define how subjects should be treated and how subjects should be taken care of. This really came out of what we all know the Nazis did under the guise of pseudo-research and the horrors that were perpetuated. So the features of the Nuremberg Code were kind of multiple. Number one, subjects had to give voluntary consent. I mean, this all seems obvious to us now, but it wasn't then. Voluntary consent, the study had to be generally for some societal, some kind of broader good. You had to create a study based on animal data and knowledge about the product. You couldn't pull it out of thin air. You had to design a study that avoided unnecessary suffering. And you couldn't do the experiment if the expected outcome was going to be death or disablement or disfigurement. The benefits had to be greater than the risks. You had the researchers had to do a certain amount of preparation and in order to provide good protection to the subjects. The researchers had to be scientifically qualified. A subject could end the study if he wanted, and the scientists could end it if it was harming the subject. Breakthrough concepts back in 1947 to organize this and kind of codify it. Okay, so what next happened that was important? Also around 1947, actually 1948, there was a drug called streptomycin. Very effective drug for tuberculosis, kind of the first TB drugs. There wasn't a lot of streptomycin around. Now, in the United States, the scientists tried to perform a kind of randomized trial about the effectiveness of streptomycin on tuberculosis. And it wasn't a very good study. It wasn't very good data. But in Britain, fortunately or unfortunately, there was a shortage of streptomycin. So a physician named Bradford Hill designed a study in which people were randomized to either get streptomycin or not. He could ethically do it because there wasn't enough streptomycin to go around anyway. I don't think you could otherwise have justified that if there had been unlimited amounts of streptomycin. The benefits of streptomycin were clearly demonstrated. This was an important randomized trial. Now, they didn't want to inject people who weren't getting streptomycin, so it wasn't exactly blinded. So people knew who was getting streptomycin and who wasn't because the streptomycin people got injected. But despite that, the data was still good. The benefits of streptomycin were well established, and the sort of trend for the future in terms of how you do drug trials was becoming a little more established. But by even as late as 1951, 45% of clinical trials didn't have a control group. I mean, what kind of data is that if you don't have a control group? Everything else is anecdotal or observational if you don't know what happens if you don't give the drug. You know, sort of consistent with that, people weren't spending a lot of money on research. Research funding in 1950 was about $160 million. Now, by 1968, it was $2.5 billion. So it exploded. In the 50s, anti-hypertension and anti-arrhythmic drugs developed. In the 60s, the TB drugs, anti-cancer drugs, heart disease drugs, oral contraceptives, all were developed as the result of clinical trials. A new science emerged called biostatistics, because you needed somebody to look at this scientific data and the number of people that were in each trial and what they were getting and decide what kind of change is statistically significant. In other words, was it more likely, was it so probable that you couldn't attribute those changes due to chance. So a whole specialty of biostatistics emerged and made possible a lot of this research. 1952, the NIH opens its research hospital and started kind of putting its stamp of approval on clinical trials around the country. Now in 1958, Senator Estes Kefauver led some hearings, some government hearings, basically complaining about the high cost of research and the poor quality of trials. And a pharmacist named Louis Lasagna said, an adequate trial is hard to find. 1958, an adequate trial is kind of hard to find. 1961, there's a drug called the thalidomide. Everybody remembers, I mean, we're all old enough to kind of remember thalidomide. Thalidomide was a sleeping pill, and it was available in Europe. And it was never approved in the United States. But it was given out to physicians to give out to patients as samples. Unproven, untested. And we all know the consequences of thalidomide and the malformed babies. So by 1961 and 1962, the FDA started to create new amendments and kind of new controls to be able to control these drugs. If you had a new drug, the company had to do what's called an IND, a new drug submission, talking about the intent to start a trial. They had to provide animal toxicity data to show that it was kind of safe in humans. You had to have manufacturing info. Was it being manufactured in a consistent manner by a reputable company with good standards? What's the chemistry background of the drug? You had to have information about the trials had to be provided to what's called an institutional review board, an IRB. IRB is a kind of an organization of people with various interests in clinical science, doctors, community members, pharmacists, people of religion, that could look at the submission, decide if the trial and the product look to be both safe and ethically okay to do a trial. And those of us doing clinical trials now live by the decisions of institutional review boards in terms of how they accept how we're going to conduct a trial. Okay, even as a result of, as late as 1962, the FDA was relying on scientific data on sort of the safety of a product, but not on evidence of efficacy. 1962, we're still not even looking at efficacy of trials. So, as this morphed along, the FDA required that, among other things, the investigators conduct the trials along with IRB approval. The investigator would personally conduct the trial. They would inform subjects that the drugs they are getting were investigational. And if there were adverse events, they had to be reported to the sponsor or to the manufacturer. In 1964, that was that famous Helsinki Declaration, which kind of worldwide sort of codified some of these general principles about how clinical trials would be designed and would be conducted. In 1970, the government came up with the drug efficacy study implementation. So finally, they began weeding out drugs in which efficacy wasn't proven. They classified drugs that are already in the market into about 6 different categories, from efficacious to non-efficacious to somewhere in between. And they started weeding out drugs, and thousands of drugs were taken off the market because they didn't fall into the efficacious class. 1996, so-called harmonization, which was when the major research countries agreed to harmonize the standards by which they conduct trials. So worldwide, clinical trials are conducted much the same manner. Now, you couldn't always conduct a clinical trial in the way you'd like to do it because sometimes circumstances forced us away from doing it. And I think in the 1990s, I think we all remember the AIDS crisis. Now, we knew about AIDS, information was being gathered, and clinical trials were being done to demonstrate whether a lot of these new medicines for AIDS would work or not work. But there was a hurry. There was an urgency. You couldn't wait. So a decision was kind of made to track these drugs. You would conduct conventional clinical trials in one group, but in another group, you would sort of, you would have much more liberal criteria to be able to provide drugs for individuals suffering from AIDS, even if the data was a little bit skewed. So that kind of brings us up to where we are now. We've got the FDA standardizing trials. We've got institutional review boards comprised of you and me and everybody else deciding on the ethics of trials. But are these randomized trials, is this the way of the future? I mean, it's clearly the way of the present. But what clinical trials do is they're kind of based on averages. In other words, you have two different groups. On average, this group does better than this group if they get this drug. On average, but people aren't averages. People are individuals. So you don't know what's going to happen with any individual, even though statistically there's evidence that one drug might be more effective than a different drug. So the future is going to be a little more personalized. We're going to be looking more at genetics. We're going to be looking more at individual genetics predispositions. We're seeing this at least in my field, in asthma and COPD, where, for example, we're looking at the genetic mapping, looking at the genes of people with COPD and asthma and tailoring therapy appropriately. And we're seeing this all across the board. So not all trials are going to be big randomized clinical trials groups. This is going to be much more individualized. But it's still going to keep happening, and there are still going to be trials, and we're going to still have to continue learning this new information. Now, if this still comes up, and I don't know it will, because laptops shut down, I may just skip this. So if it doesn't come up, what that slide is sort of talks about how specific clinical trials work now. So I thought I'd kind of walk you through the beginning of this. We'll see if this is going to come up, or it may just be too tedious. I'm not sure it's going to be necessary. I'm just going to see if this is going to work for me, but I'm not going to take very long to try this because it's sort of shut down. Now it doesn't like my password. We'll skip it. Okay, so I thought I'd give you an idea how clinical trials work in the real world. We've been doing clinical trials at my research site over near St. Jude's called California Research. We've been in clinical trials for about 20 years. I've been principal and investigator on about 150 different trials. Probably 100 different products. And they all kind of work in the same format, and we conduct them all pretty similarly. So as I've kind of outlined, a manufacturer comes up, let's say it's a new inhaler, and he thinks it's fairly effective. So what he's got to do, he's got to show some animal data that it's effective. He's got to show some chemistry data about how it works and what the biology and the pharmacokinetics are, and whether we have reasonable expectations that it might be safe. So they submit an IND, a new drug application, to the FDA. And the FDA looks at it and says, well, okay, or I want you to make these tweaks in this protocol you're submitting. And the protocol they're submitting is a design of how to do the study. How many subjects in each group? What doses are they going to get? How many visits are they going to have? How much are you going to pay the subjects? Et cetera, et cetera. So the FDA finally comes in agreement with the manufacturer. They agree on a protocol. Protocol is designed, and then ultimately the protocol or the consent form gets sent to the Institutional Review Board. So the Institutional Review Board looks at this consent and says, are the subject's rights protected? We talked about all those rights of the subjects. Are they protected? Are they covered? Are we meeting all those criteria? And of course, being in California, California has an additional Bill of Rights. So every consent has an extra page, which is California's duplication of the same set of rights. So the IRB approves that. The consent form says how much you can pay your subjects per visit. In general, we like to pay them a lot. We want people to be incentivized to do the study. The IRBs and the FDA don't want people to be incentivized. They think that if you pay too much money to somebody, they're going to be incentivized to do the study, and they don't want that. So you can only pay them so much money, and somewhere in the infinite wisdom of the IRB, they'll decide what's the right amount of money, but it's not coercive. Now, why it's a bad thing to pay people money to make them want to do the studies when you want them to do the studies, nobody understands. But this is one of the mysteries of research. It's kind of the world we live in. So they come up, so finally we come up with a consent form and a payment scheme and an outline for how the trial is going to be. How many visits are they going to have? Is it going to be 3 months? Is it going to be 6 months? How many patients are we going to have in each group? Who's getting what? And to make sure that things are randomized. So the typical place, so where do the patients come from? Sometimes they come from my practice. Sometimes you can advertise if it's not too attractive. You can't put too much money in an ad because that might be coercive. But you can advertise. They can come from other physicians. And they come in, they read the consent, and they get and then you go through a list of inclusion and exclusions. If you want people with lung disease, their lung function has to be in a certain range. They can't have cancer. They can't have pneumonia. Their blood counts have to be in a certain order. They have to have previous records. All those are the inclusion and exclusions that subjects have to meet. So when people come in and we want them to be in a trial, they got to meet these hoops. We got to turn them away. If they don't, we really have no choice. Then, typically, this is mostly kind of electronic now. If we get a subject and they're suitable, we plug them into a computer. The computer goes online, and they're electronically randomized. So they'll go back and say, well, they're going to get drug number 248. We don't know what it is because everything's packaged the same. And if it's not packaged the same, then we're required to create a whole other infrastructure of staff so you can preserve the blind. So you got to hire twice as many people on those sorts of studies to kind of preserve that blind. So anyway, finally after we do that, we meet the inclusion and exclusion. Subjects look suitable. We put them through a run-in period. So usually they go for a week or two of, in most circumstances, of just kind of stabilizing. So everybody is kind of on the same, kind of the same footing. So finally, after a couple of weeks of sweating bullets, trying to keep the subjects healthy, hoping nothing goes wrong, then we bring them back to randomize them. Back to the computer again when we submit this data, and then it tells them what drug they're going to be randomized to, and they get assigned. So finally, after several weeks, a month, subjects are finally ready to kind of receive their medicine. All the time at each of these visits, they're getting blood work, they're getting electrocardiograms, they're getting chest x-rays, as it's been outlined. Then finally, the study continues. Let's just say it's an inhaler. The subject continues on an inhaler. We don't know which one. They do an electronic diary, so they keep scores. They keep coming back to us every month or every 2 months for visits. We collect the data. And finally, somewhere down the line, at let's say 6 months, patient is done, and maybe after a year and a half, we're done with all of them. If we're really lucky and we've had a great enrollment, we get to enroll 20 or 30. And maybe along the way, 20 or 30 were screened out or had to drop out or something. So if we hit 50% of patients being able to complete the study, we're doing pretty well. All that data gets sent to the sponsor. We have to report it. If they don't hear everything, the FDA is very unhappy. So all that data has to be reported to the sponsor, bad news, good news, and then the sponsor ultimately will make some kind of decision and analyze the data, and maybe a year or two later we'll hear the results. And that's kind of how it works. As you've gathered, sort of built into the process is safety for the subjects. Protocols are designed to either keep them out if they're at risk or to kick them out if something is happening that's at risk, whether it's kind of up to us or not. And then us as investigators, you heard what our requirements are, that we have to conduct these visits and these projects ourselves. That's what we pledged to do. Then we've got to certify it, and we're on the line. Now, at any time, the FDA may decide to drop in, the nemesis of all clinical research operations, even though we understand what the FDA is doing. And the FDA, it wasn't true in 1938, but the FDA is all powerful now. So anytime the FDA wants to come, they can drop in at a moment. If you have an unhappy staff member and they drop a dime to the FDA, they can be in a minute, and they can look at anything they want. So you can have inspections that are either what's called for cause or not for cause. For cause means they got some reason to think something is fishy at your site. And we hear the stories about what happens at different research sites. Use somebody else's urine, somebody else's driver's license, somebody else's blood. I mean, any of a million things. Ranging from very benign to very serious. But those are the things that the FDA looks at, and sometimes they sort of look at them with a discerning eye and sometimes not so discerning eye. But if it's for cause, then they're looking for something, and they can drop in any time. Most of the time, though, when the FDA comes around, it's they're checking the validity of a study. So we finish a study and we enroll 20 people, and we're one of the higher enrollers on that particular study that might have 1,000 subjects. So the FDA picks us and picks a couple other sites, and they come down and do a non-for-cause visit because the drug is getting close to being put on the market. So they want to make sure our data is valid. So they can check our paper charts. How does that match up to what into the computer? Did we report all that appropriate information? If there were adverse events, did we record them? Were our conclusions valid? Did we meet all the inclusion and exclusion criteria? So that's the fear of running a research operation, is that the FDA comes in. It makes you perform pretty tightly, and it can keep you from sleeping at night, but that's really what the FDA is. So ultimately, finally, after all this, a drug gets submitted to the FDA, it gets approved, and the company is able to produce the drug. As you would have seen if this had come up, because I had a feeling this was going to die. Now it's about $2.6 billion for a drug to go to market.
That's why it's so expensive.
Well, you get an idea because of the requirements of what we have to do. Much of it goes to clinical research, and much of it goes for the cost of failed drugs. because not all the drugs succeed, but you got to pay for the ones, but you got to pay for the research on the ones that failed. So all in all, after all this, the system is actually working really well. It's staggeringly expensive. Being in the business, I become much more sympathetic, but not sympathetic to the high prices of drugs. I understand where it's coming from. I understand the costs that are involved and the requirements to produce safe drugs, because we don't want to go back to the 20s. We don't want to go back to 1980. We don't want to go back to we don't want to ethylene glycol and sulfanilamide being sent out. We want that safety. But that's the price you pay. Do the drug companies gouge on top of that? I think some of them do. But I think in general, when you understand how complex this is and the dynamics of and what requires to produce a good drug, I think you start to learn. And what we also learn is that the format is the same for all drugs. I mean, we can do a trial on COPD, we can do a trial on urinary tract infection. The protocols look the same, the format is the same, because it's all been standardized. So people are trained and they can do that. But this has only been 30 or 40 years, really, that this degree of sophistication has been applied. So anyway, I didn't really know where this talk was going to go or what was really going to happen because I hadn't done it before. But I thought it might be pretty interesting, and to give people some insight. I'm always trying to pitch clinical trials for people because I think there's a benefit for the experience. And my standard spiel when somebody comes to us and asks if they want to do it, should I do a trial? And I say, well, don't do it for the drug. You do it for the experience. Because we don't know what the drug is. I mean, the worst that is likely to happen is nothing's likely to happen. But you may benefit. And if things don't go well, you can stop. But you'll enjoy the experience. You'll learn a lot. Get a little bit of money, as much as the IRB lets you earn. And you learn a lot. And the experience is a good one. I think you're helping society as a whole. So that's my broad pitch for the research world, and I guess this is a good time to stop. Let's leave it here. Thank you all for being attentive, and if there are questions, we'll take them.
Well, I was in a National Institute of Health for Women on, of all things, calcium, and they provided me with calcium, but they didn't pay me. And they checked us, and we answered questions and so on, and I was on it for several years. And then I didn't find out I was on the placebo. Well, I mean, that's interesting.
Repeat the question.
She was in a trial with NIH, and there was a calcium trial, probably looking at osteoporosis or something like that, and didn't know what you got. Found out after a couple of years that you were on placebo. Okay. Maybe that's disappointing that you were on placebo. Maybe the alternative was that people on calcium got more kidney stones. So you don't really know whether the control arm or the treatment arm is really going to necessarily going to be better. Not all studies pay you. Some are just kind of very low level observational study. People like did it. Did you ever get this Harvard cooperative physicians form? I mean, I got that for about 6 or 7 years, or maybe longer, where I was just for a Harvard study where I was just taking a bunch of different pills just to see, sort of different supplements to see whether any of them were efficacious. By the time that it was done, it was back down to 1 pill. Didn't get paid, but I thought it was sort of contributing to general knowledge. My feeling, I always sort of felt that running a trial was is only sort of part of what you do with patients, that if all you do is do a trial, that's really not enough. I always felt you got to do some kind of value added. Take care of people in between a trial. If they need something during the trial, you take care of them. They're your people. You're responsible for them. You got to get them and you got to see them even if the protocol doesn't require. So that's why we've been doing it for a lot of years and pretty successful, because I feel kind of vested in everybody. So our subjects keep coming back year after year, not because necessarily the next drug is good, but it's just how we take care of them. So we have a coterie of people that have been doing this for 10 or 15 years, and the COPD patients will do the next one that comes along, so will the asthma patients.
How often do you have detrimental effects, or things don't work out positively?
It's really rare. How often do you have detrimental effects, or what are called adverse effects? Well, there were lots of, in a study, you got to report adverse events. That could be anything from a headache to a nosebleed to a stroke, a death. All that data kind of has to get reported. Most of the time, if you use your own investigator's judgment, you decide whether things are study drug related or not, and that's kind of unusual. So bad adverse effects are really rare. The studies just don't get that far. People are kind of cut loose earlier, and bad things that, at least in our world, we're not doing cancer chemotherapy. We're not doing higher risk drugs. We're using, inhalers and some bronchodilators and things like that are not, and the newer drugs are sort of reworked versions of older drugs. So nothing's really very controversial. So we report everything, and if we don't, we can get burned by the FDA. But it globally, adverse effects, drug-related, really, really unusual. I mean, we report adverse events all the time. The FDA got us at one time because because I didn't realize they wanted every tidbit, every headache, every heartburn. And now we just report it all, and we kind of let the sponsors decipher it. But in general, very, very few, and very few patients drop out of the studies if we're taking care of them. Not because the drugs are necessarily good. It's just because it's a learning environment where people start to understand their disease. By answering these questionnaires and everything else, they're learning about their disease. They're getting a little bit of control. They got someone who's vested in their interests. So we have dropout rates on most of these studies of a couple of %. 1 person a study.
There was one drug that they did, and they stopped the drug because it was so effective. They said it's perfectly good, and they set it out anyway. They didn't wait till the end of the trial. Yeah, I mean,
she says that that study was cut short because the benefit of the drug was so obviously effective. So a lot of studies will have kind of some checkpoint in between where the data is analyzed, and if it's overwhelmingly good or overwhelmingly bad, they'll stop the trial. If it's bad, they'll stop the trial and stop using the drug. If it's good, they don't want people to be randomized to placebo anymore because it's not fair to them. It doesn't happen for many trials, but it does.
Would you discuss a bit about the international cooperation and coordination? The Europeans have plans, it's the U.S., and I suppose China has something. And you pick up some pills, a prescription. I got some from Croatia, Germany, Israel, all over the world. So how does this work out? How do you maintain standards?
Well, let's put it, if the question is how research is conducted in terms of standards, that's kind of what this harmonization, 1996 harmonization was about, where the United States and Europe and Japan agreed to standardize the format for which all trials are done, so they can be much more international. And now we do trials all the time where the United States is one site, and we're competing for enrollment with Ukraine. So that's been standardized. Drug standards is a little bit tougher. The different countries and the different organizations have different ways of approving drugs and approve them differently. The FDA has generally been more stringent than a lot of the other countries, both rightly and wrongly. It's probably beneficial in the sense that we didn't have thalidomide approved here because the FDA moved slowly. And maybe not so beneficial because the FDA may have dragged its feet on AIDS drugs. So I think that cuts both ways. But most of the countries that do this independently, taking a little guidance from each other, but they're pretty autonomous.
About 2 years ago, there was an article in the Smithsonian about replication of studies, and it questioned the percentage of studies that are done for drugs that could be replicated. Trade secret, publish or perish, people protecting their data. Have you had
well, his question is about replication. One of the things that the FDA, I didn't really mention this, but one of the things that the FDA requires now is you don't show the benefit. It's not enough to show the benefit of drug in one trial. You got to show it in 2 trials. So the different studies can be almost identical. Same type of population you want in one group, same type of population here. Treatment arms are the same in both groups, but they're different pools and they're analyzed differently. And the FDA wants you to show that benefit in both trials. So that kind of wisely has eliminated some of that. Also, another thing I didn't quite get into is there are multiple phases to how you evaluate the safety of drugs. First, you create the drug. Then the first thing you do is you do phase 1 trials, which is when you usually get a bunch of college kids, lock them in a locked room. You give them healthy people the drug, you monitor its effects, and you do your blood work, and you establish that it's sort of a safe drug and how the levels work. Then you do phase 2, where with a relatively small number of patients, maybe 100 or a couple hundred, you start to demonstrate the dosing effects, start to demonstrate its efficacy, to establish baseline. Then you go on to phase 3, which can often be international with multiple sites and 1,000 patients. So moving to the more sophisticated level of what you were looking at in phase 2. So at the end of phase 3, a drug gets approved, and then the FDA requires that even after phase 3 and the drug's approved, that you do phase 4 trials, which continue to demonstrate the safety and efficacy of the drug and other issues on it. So drugs aren't getting approved on the basis of 1 or 2 trials. They're going through a train and have to meet these requirements at each level. And that's why that process of developing a drug can take 8 or 9 years. So it's not, so people who are, so generally when drugs are being evaluated, there are lots of trials that lead to that and tend to be pretty credible. But in the academic world, where the issue is not necessarily to produce a particular drug, whether you're sort of just doing academic data, that's a little easier to fudge. It's a little more circumscribed. People are kind of vested in this. But in general, I think in these clinical trials with big pharma, there are too many trials, too many duplications, too many different people doing the work, and too many people looking over their shoulders for there to be outright fraud. Or gross miscarriage. But it does happen that drugs get through this whole process, and finally we get on the market. People find out other things and find out it wasn't what we want, and the drug disappears. But in generally, it's continually being reevaluated. Even if the sponsors didn't want to do it, the FDA is gonna make them do it. Okay, well, we'll say two more.
In your area of expertise, when you talked about inhalers, is the placebo inert, or is it a previously an older drug as compared to the new?
Well, a placebo is a placebo. A placebo is inert. Placebo does nothing. But not, and in the perfect world, and in the perfect trial, a drug is compared to placebo. So that's the way most clinical trials work, and most manufacturers and companies want their drug to be compared to placebo because they're going to show results. But in some trials, it's not ethical. You're doing a pneumonia trial. It's not ethical that the placebo is no antibiotic. So that the control is no antibiotic. So they'll design a trial in which the control is an older or more conventionally approved antibiotic, and that acts as your control, and then you either try to demonstrate that at least is equally effective as the established drug, or it's better. So you brought up that important point, is not all studies are compared to placebos. Placebos are inert, but there are studies in which people don't get placebos. We do that. We do trials that have placebos, and I guess we got to fully disclose that. But a lot of we do with somebody already on conventional therapy, where you're doing an add-on drug that they wouldn't otherwise be able to get anyway. So there's not much downside to it. You're not depriving them of any treatment. They might not get any more than that, or they might. But those are studies that are kind of easier to praise, because you're not taking people off something. Sometimes you just have to, and the FDA demands that certain products be compared to placebo, and there's nothing you can do about it. Back there. Okay, I'm going to let you just get the last one in. After.
I happen to have a friend who is at St. Jude right now with pneumonia, which he said before, and a history of valley fever and COPD. How would she know whether you have any trial going on of anything that might benefit her? Well,
I mean, in the hospital, really nothing. Now, we did a bunch of trials at St. Jude's. I guess 2007, 2008. In fact, we did a couple of community-acquired pneumonia trials at St. Jude's. We enrolled about 40 people, about 40 people in one of them. It's much harder to do any in-hospital trials on anybody anymore, because the bureaucracy is so dense and so thick, and there's so many administrative and bureaucratic levels that in-hospital trials outside of dedicated research centers like NIH are just getting harder and harder to do. Now, out of the hospital, you never know, but one place you look at is just in general, you want to know what's out there, just go to clinicaltrials.gov, and it kind of lists everything that's out there and geared toward possible subjects. That covers pretty wide range. The other thing to look at is NIH also separately lists a bunch of trials that it's doing around the country. Okay, you had one.
Well, different people have different immune systems. So how do you standardize?
Well, I mean, I think how do people? Thanks, Joe. How do you standardize for people that have different immune systems? Well, I guess you kind of depend. It kind of depends on the study. If it's some kind of infectious study, what you make sure is that everybody's blood count is within a reasonable, acceptable range, that they don't have some kind of active disease, that their chest X-rays are okay. So kind of depending on the study, you can go into that as deeply as you want. But in general, the idea is kind of what I was saying, is that trials are based on average, meaning you get a bunch of people with different immune systems, but if you get enough of them and you compare them to another group using the same standards, then their mix is going to be about the same. So even if you can't compare 1 to 1, you can say on average, this people with these conditions are like these people with these conditions. So it's fair that if we see a difference in the drug we're treating them with, that it's due to the drug and not idiosyncrasies between people. That's why we need those biostatisticians. Okay, so I think that's it. Oh, Joe's got a question.
How do you recruit? Suppose you're going to do a trial on a certain drug. How do you get patients involved in that at your bulletin?
Well, that's the, you know.
How do you get the word out?
Yeah, there are two things that you need to run a successful research site. You need trials, which is hard enough to get because it's competitive, and you need subjects. My sort of feeling, because we've been doing it for a long time, is I sort of have a vested interest in everybody. So if someone finishes a trial, they don't have insurance, whatever it is, they've finished their trial, I make sure I take care of them anyway. If I got to give them samples, if I got to see them in the office, I keep them around. So that's both my good, wholesome nature, and it's my self-interest, because if I take care of them, they're going to be around and they're going to be available and they're going to be willing to do the next thing. So I think that's mutual benefit, and after we've done that enough years, there's a pretty good stable of people that are loyal to us. I mean, we started a new asthma trial on New Year's, and pretty much called up everybody we knew, and we've got 20 people randomized now, and we're the highest enroller in the country, and 16 or 17 already belonged to us. So we knew them, they were friends, we had taken care of them, they were kind of either we call us or they call them. And secondly, you can do advertising within certain scope. The Institutional Review Board, in its all wisdom, has to look at advertising. So you can say, we've got an asthma study, subjects will be compensated for time and travel, but you can't say asthma patients come in, you can earn $2,000. So you have to work within fines. So you can do newspaper, you can do radio. Now there's a lot of internet advertising. So there are companies that specialize in advertising on various social media, and particularly for patients like young asthma patients who just won't read anything, but they'll be on the internet and they'll answer. So we get that. Sometimes other physicians and other centers will be helpful if the light bulb goes on for them and they kind of understand what we're trying to do. If the light bulb doesn't go on, it'll never go on. So those are the three sources. But basically, we try to keep it, for us, we just try to keep everybody close to the vest and make sure we call them if we don't have a trial. I mean, we hadn't had a COPD trial for about a year. We finally got one. Patients are calling us. I mean, they're all ready to come back in and be taken care of.
Okay, great. I found this program very fascinating. It's a key issue in our healthcare system today, and I'm glad you all got to hear, especially from Dr. Pearle, who is so experienced and well recognized as a trial expert.
And good
so
job.
thank you very much.
Right.
And great job. And then,